many of the obstacles to discovery. First, it's clearly a non-invasive, wireless form of communication. And second, just as in a radio broadcast, you can communicate with many receivers at once. You don't need to know where these receivers are. And it doesn't matter if these receivers move. Just think of the stereo in your car. It gets even better. For it turns out that we can fabricate the receivers out of materials that are encoded in DNA. So each nerve cell with the right genetic makeup will spontaneously produce a receiver that allows us to control its function. I hope you'll appreciate the beautiful simplicity of this concept. There's no high-tech gizmos here, just biology revealed through biology. Now let's take a look at these miraculous receivers up close. As we zoom in on one of these purple neurons, we see that its outer membrane is studded with microscopic pores. Pores like these conduct electrical current and are responsible for all the communication in the nervous system. But these pores here are special. They are coupled to light receptors, similar to the ones in your eyes. Whenever a flash of light hits the receptor, the pore opens and electrical current is switched on, and the neuron fires electrical impulses. Because the light-activated pore is encoded in DNA, we can achieve incredible precision. This is because, although each cell in our bodies contains the same set of genes, different mixes of genes get turned on and off in different cells. We can exploit this to make sure that only some neurons contain our light-activated pore and others don't. So in this cartoon, the bluish-white cell in the upper left corner does not respond to light because it lacks the light-activated pore. The approach works so well that we can write purely artificial messages directly to the brain. In this example, each electrical impulse, each deflection on the trace is caused by a brief pulse of light. And the approach, of course, also works in moving, behaving animals. This is the first ever such experiment, sort of the optical equivalent of Galvani's. It was done 6 or 7 years ago by my then graduate student, Susanna Lima. Susanna had engineered the fruit fly on the left so that just 2 out of the 200,000 cells in its brain expressed the light-activated pore. You're familiar with these cells because they are the ones that frustrate you when you try to swat the fly. They trigger the escape reflex that makes the fly jump into the air and fly away whenever you move your hand in position. And you can see here that the flash of light has exactly the same effect. The animal jumps, it spreads its wings, it vibrates them, but it can't actually take off because the fly is sandwiched between 2 glass plates. Now, to make sure that this was no reaction of the fly to a flash it could see, Susanna did a simple but brutally effective experiment. She cut the heads off of our flies. These headless bodies can live for about a day, but they don't do much. They just stand around and groom excessively. So it seems that the only trait that survives decapitation is vanity. Anyway, as you'll see in a moment, Susanna was able to turn on the flight motor of what's the equivalent of the spinal cord of these flies and get some of the headless bodies to actually take off and fly away. They didn't get very far, obviously. Since we took these first steps, the field of optogenetics has exploded, and there's now hundreds of labs using these approaches. And we've come a long way since Galvani's and Susanna's first successes in making animals twitch or jump. We can now actually interfere with their psychology in rather profound ways, as I'll show you in my last example, which is directed at a familiar question. Life is a string of choices, creating a constant pressure to decide what to do next. We cope with this pressure by having brains, and within our brains, decision-making centers that I've called here the actor. The actor implements a policy that takes into account the state of the environment and the context in which we operate. Our actions change the environment or the context, and these changes are then fed back into the decision loop. Now, to put some neurobiological meat on this abstract model, we constructed a simple one-dimensional world for our favorite subjects, fruit flies. Each chamber